All right, it's 2, so we will get started. I should probably plug in my tablet. So for test 3, the grades are up. And I've dropped your lowest test. So if test 3 was your worst, that's been updated. You shouldn't be able to see test 3 yet, though, like check your answers. So I will have to do that and let you have access to it. And when you look over it, if you have questions of why I scored something, please let me know. So that's test 3. Remember, Friday, no live lecture. So just look for the recording in the course content. I will be making that recording when we're finished here today. So yeah, those are the only major announcements so far. So let's get right into our material then. And on Friday, where we left off was looking at the action potential, right? And we talked about how sodium and potassium channels will work together to propagate a nerve signal. And this is a good review question to look back on that. Basically, go back to Friday's lecture and it just goes over that whole spiel I went on is this. But I do want to go into the actual channel itself. And the main channel we're going to talk about is the potassium voltage-gated channel in the nervous system. And it's called a voltage-gated channel because it opens and closes based on the charge of the cell. So if you remember, when a nerve impulse happens, sodium, if this is cytoplasm, sodium goes into the cell, making the cell positive. Then potassium leaves the cell to balance out that charge and becomes negative again, and then the channels close. So this potassium channel is being opened because of charge. So the question is, how does this happen? And when we look at the amino acids in our voltage-gated channel, and this is a cartoon of it, you have 6 transmembrane helices. However, this 2D image really doesn't show you what it looks like. These helixes are like forming a funnel almost. So they're not just like linear in the membrane. This is the actual image where they're in a circle, allowing the potassium to go right through the center here. All right. So in helix 4 of this channel, you have conserved arginine residues. You have 4 conserved arginine residues. And remember, arginine is a positive amino acid, so it has positive charge. When sodium flows into the cell, this positive charge will cause the arginines to want to move away because like repels like. So this S4, the arginines, will move towards the outside of the cell in the membrane. So they'll this whole channel will shift. And it's kind of being shown here in red. You can kind of see it shifting. If this is the, if this is the cytoplasm, this is the outside, you can kind of see it move towards the outside of the cell a little bit compared to its previous position. When this happens, this causes the whole channel to change conformation, opening up the helix and allowing potassium to flow out and neutralizing the charge from the sodium flowing in. So that's how this channel can actually sense and open based on charge. Once potassium has left the cell, and the cell becomes negative again, so let me change that up. Once the cell becomes negative again, this helix will be attracted to the inside of the cell again. S4 will go back to its closed position, and the potassium channel closes. So that is how structurally this channel is a voltagegated channel. This is how we can sense, what is the charge on the inside of the cell? Okay, based on that charge, am I open or am I closed? It's because of 4 conserved arginine residues in helix 4. However, when we study the potassium channel, we notice that it closes way faster than what would be predicted if it's based solely on this helix 4. And that's because on the N-terminus of this channel, we have what's called the inactivation ball. And what the inactivation ball is, it's an area of hydrophobic amino acids. So you got some hydrophobic amino acids here. And once the pore opens, what happens is that this ball unfolds and will bind inside the pore, because inside the pore you also have hydrophobic amino acids. So this ball swings into the pore and shuts it off very quickly. Otherwise, you have the potential to lose a lot more potassium than you would need to do. And that would be very bad for the cell, as the cell does need potassium to function. And we know it works this way because if you chop off the N-terminus, it takes a much longer time for this channel to close, because then it would be solely reliable on S4 to close. So really the way to think about it is that S4 opens the channel, this inactivation ball will swing in and close it, and then when S4 goes back, it can just reset, and this pore will start to close, kicking the inactivation ball out, and it can be reset for another nerve impulse at that point. And although we're studying just the potassium channel here, when we look at other channels such as sodium and calcium, they kind of have the same mechanism going on. So it's similar structure there. So any questions about our potassium channel to begin with? All right, now we move on. So we're going to continue on our studies of transporters. Next one we're going to look at is extremely common. It's called an aquaporin, and an aquaporin moves water. Again, water is polar, so it's not going to be able to go through the cell wall itself. It has to travel through aquaporins. And we're going to look at AQP1, which is the best studied aquaporin. And this is a glycoprotein, which means it has a sugar attached to it. Homotetrameric. What that means is that it's made out of 4 different subunits, 4 different proteins that are the same. So 4 copies of the same proteins come together to make one functioning aquaporin. And when we do this, we have 6 transmembrane alpha helices and 2 other helices that go right through the bilayer. Look at it as in the picture in the bottom left. It kind of looks like an hourglass. Can kind of imagine that. And what's interesting about this is when we look at the pore, we have those hydrophobic groups, which you would not necessarily expect. If you're transporting water, you might think, well, you need groups that will hydrogen bond your water. And you do. You have arginines and histidines, and also backbone groups of your amino acids that will hydrogen bond the water and help them pass through the channel. However, the idea here with hydrophobic amino acids is to just move water through the channel faster. You know, I could think of a situation where if you didn't have these hydrophobic amino acids, water would just go in the channel and hydrogen bond and just stay there for a little bit, because why would water want to move away? It's happy with hydrogen bonding. So these hydrophobic groups are kind of a way to just keep the water going through the aquaporin. You don't want to just stop there. You want to keep on flowing. What is important, though, is that we have two asparagines here in this image. You can see we have a water trap behind here. These residues help prevent what's called proton hopping. I talked about this in the video, but I'll also cover it here because it comes up a lot in biochemistry. Now, what proton hopping is that when water is H3O plus, an individual water molecule doesn't stay as H3O plus for very long. What it does is that it'll pass its hydrogen off to other oxygens, or other water molecules rather, because being a positive oxygen is very unfavorable. So it will pass off that oxygen to another water, which will pass off that oxygen to another water, so on and so forth. And what you see is that hydrogens can appear to move incredibly fast inside of the cell because you have this proton hopping. They're going way faster than they could through diffusion, for example. And so what these central asparagines are doing is that they're separating water, this water molecule, from the other neighboring water molecules. If these asparagines weren't here, for example, then you could have a clean chain of water molecules going through the aquaporin, and you could export hydrogens inside or outside the cell incredibly fast. For a prokaryotic organism, this would be really bad because prokaryotes, they don't have mitochondria, right? So when they want to do oxidative phosphorylation, they use the inner membrane space in the cytoplasm as a way to have their hydrogens be imbalanced. Well, if hydrogens could flow through an aquaporin, you would ruin ATP synthase, and your prokaryote's not going to make ATP, and they'll die pretty fast because the protons would just naturally go through the aquaporin through proton hopping. However, since we have these asparagines, we're stopping proton hopping, and we can stop hydrogens coming through this process. So we make sure only water goes through and we aren't transporting hydrogens. Right, so questions about the aquaporin there? Right. So this question, question 6, it's like a question that I asked on the test that many of you did not do well on. So on the test, I asked, tell me structurally why hemoglobin has positive cooperativity. And I think only like 3 or 4 of you answered that like sufficiently. So in biochemistry, now that we're looking at proteins and we're talking about the structure of proteins, I want you to be able to look at the structure and tell me how structurally that protein is able to do its function. So here we have the potassium channel and the aquaporin channel. So just just so we're all on the same page, I will go ahead and answer this, right? So voltage-gated channel opens, lets potassium flow out of the cell due to a change in voltage. How does the structure of the channel relate to this? Well, helix 4 has 4 conserved arginines. They move when cell becomes positive charge, which opens the channel. All right, so that just explains how this channel can open up because of positive charge. And then to be completely sufficient, you would want to go on and say, then once the inside of the cell becomes negative, the positive charges go back towards the cell, closing the pore. And it also has that inactivation ball to do that as well. So that's the potassium gated channel there. So I want you to practice the aquaporin one. And instead of just giving you the answer like I normally do, I'm gonna not give the answer for that one. And if you have questions about it, I want you to come in, talk to me, email me about that. As I think I've only gotten like one person, maybe 2 people to email me about questions during this whole course, which is incredibly low and maybe related to why the test scores are way lower this semester than any other semester, even when I did it in the fall. So as a way for you to come and ask me questions, I'm going to leave that one open for you and try to solve. So questions about that? All right. So the last transporter we're going to talk for this slideshow before we move on to the slides I made for today is the glucose transporter. You have various different glucose transporters in your body. They're generally called GLUT for glucose transporters. So we're just looking at GLUT1 here. Don't worry about GLUT1, GLUT2, GLUT3, GLUT4, what any of those mean. If you take biochemistry 2, then we'll talk about the differences in them. But let's just look at the basic way this transporter works. So GLUT1 transports glucose in 4 different steps. And it's different than the other transporters we were talking about previously, where we have a channel and then the molecule just binds its way through the channel. Here, we're actually changing the entire channel itself, doing a conformational change. So the 4 steps are, you have glucose on the outside of the cell, and it binds to the transporter. And all of these are reversible. GLUT1 can be used to bring glucose in or kick it out of the cell. Once you have glucose bound, you do a conformational change. So you can see at the beginning of GLUT, it's only open to one side of the cell. When glucose binds, the channel changes, and now we're open to the other side of the cell, right? Once we're open, the glucose is free to dissociate. And I do want to point out, you can bind here and just immediately unbind as well. But you can also go through this conformational change. Now you're open, the glucose can go into the cell, and then you reopen up. And the reason why we go through the trouble of having only one side open at a time is that we don't want any other molecule to slip through, right? We don't want water or any ions to be transported. As we have seen, concentrations of things matter a lot, and we want to control what goes in and out of the cell. So by having GLUT open and closed, we are making sure nothing slips by, and we are only transporting glucose. So those are the 4 steps of GLUT. Glucose binds, change conformation, glucose dissociates, go back to the original conformation. I believe, yep, that's the last slide. So let me switch slides here. Go to the 14th. All right, and we are still talking about transporters here, but now we're going to move into active transporters. So let me just rewrite some stuff here that's cut off on your screen, I'm sure. And we're going to talk about active transport by the sodium potassium ATPase. So active transport, whenever you see active transport, you should immediately think, oh, we're using energy. That's usually ATP. Some also use GTP. But as you can see, this is an ATPase, which means we're using ATP here. So let's go through the 4 steps of our, well, 6 steps actually, of our ATPase here. And we're going to start in the top left. And so some things that are confusing about this picture, even though there's arrows moving, we're just talking about a transporter that sits in the membrane. The membrane's not like physically moving inside and outside of the cell, right? Think of this diagram just like the GLUT transporter we were talking about, where it just sits inside the cell, but changes conformation. And you have 2 different conformations. You have conformation E1 and conformation E2. E1 binds sodium on the inside of the cell. E2 binds potassium on the outside of the cell. All right, so we're going to start in the top left, where we're in the E1 form. So we're going to bind sodium, and we have ATP already bound. And every time we do this, we're going to bind 3 sodium in E1 or 2 potassium in E2. So 3 sodium will bind to our enzyme. So we have 3 sodium. And we also have ATP. After that, we are going to hydrolyze our ATP. This magnesium here is just a cofactor. And it's just going to help us split that ATP. We're going to split it into ADP plus phosphate. So the ADP is going to fly away, but we're going to have a phosphate, I'm going to call that PI, that is now attached to our enzyme. This high energy form of our enzyme will allow sodium to go inside the cell. So sodium is transported through, or sorry, outside of the cell, And when that happens, we have a conformational change. We have the enzyme still have phosphate, but now it's in the E2 form. Once it's in the E2 form, it will bind 2 potassium on the outside of the cell and then remove the phosphate. Once the phosphate is removed, the potassium can go through the channel and be transported inside the cell. When that happens, the enzyme goes back to its original conformation, E1. And it also has ATP bound to it. So let me just draw little pictures of that again over each image to make sure we have it. So we have E1 with ATP bound. Then we bind 3 sodiums on the outside, ATP still bound. Then we hydrolyze, so we're E1, just a phosphate now with 3 sodiums. And those 3 sodiums go through. Once they go through, we have changed conformations, and now we're E2P. E2P can bind to potassiums. Once we lose the phosphate, the potassiums can go through. And now we're back at the start. So that's the mechanism for our sodium potassium ATPase there. So questions about that? All right, if not, we can move on to the next slide, which I believe is a question slide. And this is, do you think sodium moves faster through a pump or a transporter? And why? And just a little hint, what we just looked at was a transporter. On the previous slideshow, those were pumps. And so when we think about it in those terms, pumps way faster. Because the difference is that a pump is mainly going to be a channel. It doesn't require energy or conformational changes, and ions can just flow through. Transporter will usually have some kind of conformational change, and usually require energy. So that conformational change takes time and it's slow. So a pump is much, much, much faster, but a transporter does have a good deal of control to go with it. So the previous ATPase we looked at was sodium potassium. Now let's look at another type of ATPase called the calcium ATPase. And this, as we'll see, is basically the same mechanism that we just looked at. Let me write what is hidden here. So this says 2 to 3 hydrogens. This says H2O. This says phosphate. All right, so calcium is very important because a release of calcium does a lot of different things. We already talked about back in like chapter 6 how our actin is covered in proteins, topomyosin. And these proteins are only released when bound to calcium. So a release of calcium triggers muscle contractions. Calcium can also do our nerve impulses that we looked at. And in Biochem 2, one of the things we talk about is how calcium can signal a lot of different metabolic processes. And one I just have listed there is glycogen breakdown. And the difference in calcium between the cell and the outside, the extracellular space, is huge, right? So listed there, calcium inside the cell, 0.1 micromolar. Outside, 1,500. And so if you had a pump, those two levels would equilibrate super fast, and you would have all these different reactions happening. And so we don't want that to happen. And so to help control the flow of calcium, we use a calcium ATPase. Like the sodium potassium ATPase, we are pumping molecules opposite directions. Right here, we are pumping calcium outside the cell against the gradient, and we're bringing hydrogens inside the cell. And again, it's a very similar mechanism. And even though this picture on the bottom left there is showing like arrows moving, again, here's the protein inside and this is the membrane where the M is. This thing isn't moving. It's just changing conformations between E1 and E2, which is shown here. So E1's in purple, E2's in green. And the biggest change happens in the part that's not in the membrane. We just have this huge movement. Oh, there's 4' of all of these subunits. So a big movement, and that whole conformational change, 2 goes all the way over here. That conformational change is due to ATP. So let's take a look at this. So we're going to start in the E1 conformation again with ATP bound, just like in the sodium potassium ATPase. And in this condition, we'll bind 2 calcium on the inside of the cell, and this will hydrolyze that ATP to make a high energy E1 phosphate, just like in our previous mechanism. When we do that, we can pump the calcium out, and we're left with an E2 phosphate, just like in the previous mechanism. An E2 phosphate can bind 2 to 3 hydrogens, like so. Once we have our hydrogens bound, we're going to lose our phosphate. And then our hydrogens can go through inside the cell. This brings us back to E1, and it'll bind ATP again and ready to go. So it's almost like 100% the same as the sodium potassium ATPase, but here instead we are moving calcium and hydrogens. So questions about our hydrogen calcium pump here, or a calcium ATPase. Right. Here's a math question. We can just skip this. We can skip the pH. Don't worry about that type of calculation. But I do want you to make sure that you know like which direction, like this kind of transporter would work. So if we look at the question, So cells in the stomach secrete HCl at a concentration of 0.15 molar. Protons are pumped out by a hydrogen potassium antiport. An antiport is what we've been talking about, by the way. Antiports will transport things in opposite directions. While a sym transports something in the same direction. So draw a diagram showing what direction these are pumping these ions based on this description here. So I'll give you like a minute to figure this out, and then I will draw it for you. So try and figure out what directions are these going. If I have a cell, if this is a stomach cell. Right, so this is a cell lining your stomach. What direction is the antiport working, and what direction is the symport working? So I'll give you a minute to do that. All right, so if this is a stomach cell, and up here is just stomach fluid. If I'm pumping HCl into our stomach fluid, so let's look at our antiporter. So hydrogen must go out of this cell. All potassium goes inside because that's what an antiporter is. And then symporter, I want to put chlorine. So potassium and chlorine. So here, it's interesting. We bring potassium in, then we just pump it right up. So no net movement of potassium. It's just along for the ride. But here we are secreting our HCl into the stomach fluid. So that's the directions we're going. Make sure you know what an antiport is, what a sym transport is. And the last one is a uniport. Right, that was our glut, where we're just transporting one thing. So we got a uniport, antiport, sym transport. Make sure you know those definitions. And if you don't watch my videos, you would not know that. So questions about that? Right. So we are continuing talking about transporters. And the next one we have is ABC. And these stand for ATP binding cassette. ABC. And what these transporters are is that they bind ATP to move a molecule across the cell, right? So you have these NBDs, or nucleotide binding domains. These are consistent to ATP transporters. It's what makes an ATP and an ABC and ABC. These things will transport a lot of different things. You have a lot of different transporters, 48 in humans. So like ion, sugars, amino acids, polar, nonpolar substances. And it's really annoying for cancer cells when you try to kill a cancer cell using drugs. One way they can get around that is that they can overexpress a certain ABC transporter called P-glycoprotein. And what P-glycoprotein does is that it pumps amphiphilic things out of the cell. If you remember, amphiphilic means it has polar and nonpolar groups. Polar. And drugs are mainly amphiphilic. If you look at structures of drugs, a lot of times they are amphiphilic. So if you try to kill a cancer by taking in certain drugs, they'll just pump those right back out and you'll kill normal cells. Excuse me, I had to sneeze there. And yeah, so main thing here, make sure you know what an ABC transporter is, make sure you know what they have in common, which is the next question. So what do they have in common? Nucleotide binding domains, they can bind ATP. And one common disease that is caused by ABC transporters is cystic fibrosis. And so normally, most people have CFTR inside their lungs, which is called cystic fibrosis transmembrane conductance regulator, or CFTR. And it's an ABC transporter that transports ions, and specifically chlorine ions. And they pump chlorine ions out of the cell. Now, individuals with cystic fibrosis, for whatever reason, CFTR does not work. Now, I want to say oh, it actually has it right there. I was going to do that off memory, and then I wrote it down. 70% of cystic fibrosis is because CFTR is missing exactly one amino acid. That's it. It's missing phenylalanine 508, and then this ABC transporter doesn't work. What happens is that if you don't have chlorine go into your lungs, your mucus will start to thicken, and then you won't be able to get rid of it because it's too thick. One, this would cause trouble breathing. But what also happens is that bacteria starts to grow in this mucus and they don't get cleared out. And then you start getting infections. On the middle picture there, those are some symptoms. So sinuses, lungs, you have infections, and then a bunch of other complications, all due to one amino acid being deleted in an ABC transporter. That's it. That's all it takes. Right. Well, I'm just seeing where we're at. Okay. So I think I'm going to finish up with the glucose transporter. I'm going to do these last two slides and then we'll call it a day for today. So 2 more transporters to talk about. Next is the glucose transporter. And the glucose transporter is very interesting. And this is in your guts, in your intestinal epithelium, if you want to be technical about it. And we already talked about glucose transporter here. It brings in glucose. However, the glucose transporter itself doesn't use ATP. It uses what's called secondary active transport. That is, it uses ATP from a different transport system to do its own function. What do I mean by that? So if we look at our transport system here, we have a symport, a sodium glucose symport, that's bringing glucose into the cell. This isn't using any energy. However, if we didn't have a way to get rid of this sodium would build up and this symport would break down. So to keep sodium low, we pump it out of the cell using our antiport here, our sodium potassium ATPase. So this pumps sodium out, brings potassium in, which keeps the concentration of sodium low inside the cell, which is favorable for when this symport is working. So we can bring sodium and glucose in at the same time, and that free energy is coming from pumping sodium out later. So that's why this is called secondary active transport. We are moving glucose across into the cell using the free energy of a sodium potassium ATPase. And this should say salt, not sail. And so one thing to do is if you have diarrhea and you're losing a lot of water and salts, you should give people glucose with salt water, and it will help them absorb salts into their body that they're losing. So that's just a practical application of here, because glucose will help bring that sodium in through this symport, and you can absorb it into your body to help protect against what you're losing. So that's the glucose transport, and that's what secondary active transport means. And the last transporter we're going to look at today is co-transport through lactose permease. Are we actually going to look at this? You know what? Let's just say, for the sake of time, this is more or less exactly the same as our sodium potassium ATPase and our calcium ATPase. And so I really don't want to just talk about the same mechanism again. Hopefully you got the point by now. So when it comes to the test, I will ask you questions about the sodium potassium ATPase and the calcium ATPase. We can skip the lactose permease, which is more or less the same thing, but you don't use ATP here. So yeah, if you're really interested in that, feel free to read this slide. Feel free to look in your book. Otherwise, I think this is a good time to stop. So just remember, I will be putting up the lecture videos for what we're covering on Friday. I'll be doing that in about an hour here. I'll probably record, let my voice recover. Yeah, so don't come here at 2 o'clock on Wednesday. I won't be here. The video will be up. Otherwise, I'll put homework up like always. And hope to see you on next week, Monday. Take care.